In this video we will discuss about a new paper or a different paper where we will discuss about how this matrix stiffness affects this Hep-2 laryngeal cancer cells, morphology and its growth. So they checked this cell growth and morphology on two different stiffness, and the hydrogel that they used for this 1 kilopascal and 8 kilopascals is made up of polyacrylamide gel, and they coated it with collagen. So they used a 1 kilopascal for soft stiffness and 8 kilopascal for stiffer hydrogel. So in the soft hydrogel, when they culture the cells after 24 hours, they are able to see that cells are still in the rounded form. Whereas in the 8 kilopascals compared to the 1 kilopascals, cells stretched out. And this trend continues, that with increasing the stiffness, they are able to see cells are more stiffed out. So after that, they check in order to confirm that there is an effect on the morphology. They checked how much area is occupied by the cells on different stiffness, and they are able to see that the adhesive cell area is more for the 8 kilopascal, as it is expected since cells are stretched out in the 8 kilopascals compared to the 1 kilopascals. And thereafter they count the number of cells at different time intervals, and they are able to see that with increase in the time there is increase in the number of cells that are seeded. They increase the number of cells in the stiff hydrogel compared to the soft hydrogels, and this could indicate that there is a growth rate of the cells is high in the 8 kilopascals compared to the 1 kilopascals, or the cells are more proliferative in nature with increase in the stiffness. And these things they have further validated using the proliferation marker, that is Ki67, and we can see that at the lower or at the soft matrices, the Ki67 positive cells are less, and with increase in the matrices stiffness, we are able to see the Ki67 positive cells increases. And these things for the 1 kilopascal and 8 kilopascals, they have quantified in the right hand side of this panel D, where we can see that for 1 kilopascals Ki67 markers is around slightly more than 30%, but for the 8 kilopascals it is more than or close to 70%. Afterwards, they also checked the gene expression levels of the proliferation affected markers, such as proliferation affected markers such as cyclin D1 and cyclin D3. It is related to the G1 cell cycle stage, and they can see that with increase in the stiffness, this proliferation associated genes, that is cyclin D1 and cyclin D3, are also increased. And these things they have also quantified on the right hand panel E after these Western blot signals, where they see that the increase is statistically significant for both cyclin D1 and cyclin D3 on the 8 kilopascals compared to the 1 kilopascals. So from this results, they come to a point that with increase in the stiffness, it not only changes the morphology of the cells, but it will also leads to the changes in the intercellular signaling in a way that it will promote more proliferations. It will promote proliferation or the increased cell growth rate compared to the soft matrices. And these things they are validated by multiple ways. For example, the proliferation rate is validated by cell counting and also by the proliferation markers. And at the gene levels, they validated it with this increase in the cyclin D1 and cyclin D3, which is associated with the G1 cell cycle step. And for the morphology, they checked it in the bright field images and then confirm it using the quantified adhesive cell area, where they can see that the effect of morphology is statistically significant. And these things they have done on these Hep-2 laryngeal cancer cells. I hope you understood how they checked the effect of the matrices on this cell morphology and growth. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.